Hi, and welcome. In this final module, we will discuss data extraction and how you may want to use your data collected to then perform a meta-analysis. We will build on the previous lectures and follow on from what happens once you have screened your studies for inclusion and have decided which studies are of relevance to your research question. Once you have selected studies for inclusion in your systematic review, you can extract relevant data you want to present in your summary. Depending on what you're interested in, what data you are able to collect, you can choose to summarize your data qualitatively by describing the field in general, and if you have enough quantitative data and it is appropriate, you may also want to perform a meta-analysis. So you may want to extract data like where a study has reported randomizing the animals to an experimental group, or performed a study blinded to these group allocation. You may also want to record information about the characteristics of the samples being used, the parameters of any interventions given. Depending on if you plan to perform a meta-analysis, you may also want to extract details about the outcome that was measured, time at which this was done, and any other relevant information. To perform a meta-analysis, you will need to make sure you extract an aggregate value of effect, so a mean, a median, or event data, and an estimate of measure of variance like SD or SEM, and the number of animals that the outcome was assessed in. It's important that you pre-specify these variables and set out what you will extract from studies exactly in your protocol at the beginning of your review. To reduce bias and human error, we advise that you have more than 2 independent researchers extract data. And to deal with any disagreements, the independent extractors can discuss discrepancies with input from an additional review author if necessary. At the beginning, what is a meta-analysis? So meta-analysis is a quantitative statistical technique to combine the results from multiple studies. One of the benefits of performing meta-analyses is that it can increase the power and precision in estimating overall efficacy of an experimental setup, more so than if you were basing your conclusions on simply a single study. This is especially useful when findings are numerous and inconsistent with each other across different samples. Performing a meta-analysis should be goal-oriented, and therefore it's important to know what you want to do before you start. As I mentioned previously, you should pre-plan your analyses to minimize bias introduced later on, once you have seen what the data actually looks like. Your plans for analyses should be detailed within your protocol and made openly available to others before you start the review process. You may want to upgrade your protocol as you go along, but these changes should be justified and recorded. Important consideration to take into account before going ahead with a meta-analysis is whether it is actually appropriate to do one, considering the data you have. First of all, it is important to make sure that you have adequate power to detect the effect you are interested in. So if you have a really low number of comparisons in your dataset, then it is probably not a good idea to do a meta-analysis, as this won't be able to detect a meaningful effect of your intervention of interest. Secondly, if what you are trying to lump together is wildly different, it may not be appropriate to include in the same analyses. You can and should perform separate analyses if, biologically speaking, you are looking at very different things. For example, it is not advised to lump together behavioral outcomes and biochemical measurements. More simply, performing a meta-analysis requires some key data, and if these are missing, you won't be able to include these data in your quantitative summary. These values may in some cases be imputed, but in many cases you will just have to exclude the comparisons with missing information from your analyses. If you want to delve deeper into what factors influence your analyses, then there is also an issue of power here. So if you have a dataset where you have a large list of treatment drugs, for example, where many of them only contribute a handful of comparisons to the entire dataset, you might have to consider grouping some of these drugs based on some sort of biological reasoning to increase the power of the data in each of your categories. This is also difficult and can be unreliable if you have severely unbalanced data. An example of this would be if you had a variable where a large proportion of your studies had one answer and only a few studies had an alternative answer. This would mean your data for that variable were unbalanced, and any conclusions drawn about its influence on the outcome of interest would be unreliable. The first stage of meta-analysis involves calculating a summary statistic for each of your comparisons to describe the observed effect, called the effect size. This is essentially the difference in outcome that is being measured between two groups of animals. By comparisons, I mean here all instances where an outcome of interest in a cohort of experimental animals is presented alongside the outcome of interest in a control cohort and is being compared to determine the effect of the experimental intervention. Depending on if you are characterizing a model or if you are looking at the efficacy of a treatment in an existing model of human condition, you will have to calculate a summary statistic for each of your comparisons between a control and experimental cohort. If reported, it is sometimes also useful to extract a naive animal's performance too, as this will help to determine what method you can use to calculate your overall summary statistic. Depending on what kind of data you have, you will have to use a different method to calculate your effect size for each of your comparisons. If you have continuous data, you may be able to perform a raw mean difference calculation if your outcomes are reported on a meaningful scale, and all comparisons in your review use this scale. If your scales differ, but you can still infer how sham-naive, unleashed animal would perform, you can use a normalized mean difference approach to calculating your effect size. A standardized mean difference is also often used when you are combining lots of different outcome measures, and it doesn't require for scales of measurement to be the same and standardizes these so they can be meaningfully combined in your meta-analysis. For dichotomous data, you may calculate an odds ratio, and where data are presented as median survival data, you will have to approach this differently again. One thing you have to take into account when calculating an effect size is the direction of effect of the experimental intervention you are interested in on the outcome. So the scale or measurement you are to measure your outcome with, does a higher score mean the outcome we are seeing is better or is it worse? And depending on what you are comparing, what your outcome measure represents, you will have to apply a correction factor when calculating your observed effect size. Where multiple similar outcomes are reported from the same cohort of animals, we want to avoid double counting these cohorts of animals and include them multiple times in our meta-analysis, as this will create a unit of analysis error due to the unaddressed correlation between the effects from these different comparisons. This can be problematic because this runs the risk of drawing artificially inflated or erroneous conclusions about the true effect size of a model or an intervention depending on the types of comparisons we are looking at. We must essentially choose either to extract a single outcome or to combine more than one outcome. If we wanted to use a single outcome, we might select the smallest effect size or have a hierarchy of preferred outcome measures, or only include data for one specific outcome measure. If we want to combine outcomes, we can extract all outcomes and nest biologically relevant ones using a global measure for the behavioral outcome in that comparison. To nest outcomes, we sum weighted outcomes from each comparison and divide them by the sum of the weights. And the standard error of this effect size is given by the square root of the number of comparisons divided by the sum of the weights. Before we can calculate our pooled effect, also known as a global estimate, we need to consider that not all studies will contribute to this effect equally. For example, if all studies in the analysis were equally precise, we could simply compute the mean of the effect sizes when calculating an overall effect. However, in a meta-analysis, it is usual to attribute different weights to each study in order to reflect relative contributions of individual studies to the total effect estimate. We do this according to the precision of each comparison, so that more precise comparisons are assigned a greater weight in the calculation of the pooled effect size. We recommend using the inverse variance method, where individual effect sizes are multiplied by the inverse of their squared standard error, or SE. This gives a weighted effect size, where ES is the individual effect size and W is the weight. For median survival or the time to event data, we weight effect sizes according to the total number of animals, which is calculated from the true number of control animals plus the number of treated animals in that comparison. We can now finally begin to pool our effect sizes to calculate an overall effect size. There are two methods to doing this, so let's start with the fixed effect model. Under the fixed effect model, we assume that there is one true effect size which is shared by all the included studies. It follows that the combined effect size, or the global estimate, is our estimate of this common effect size. The only source of error in this model, and the reason why the comparisons we include differ, is due to sampling error. The alternative to this is the random effects model. Under the random effects model, we allow the true underlying effect to vary from study to study instead of assuming that there is one true effect. We assume that the underlying effects are normally distributed around some average effect of these different underlying effects. So for example, we say that the effect size might be a little higher if the patients are older, the outcome is slightly different in rats versus mice, and if the study used a slightly more intensive or longer variant of the intervention, then this would also give a different underlying effect. The studies included in your meta-analysis are assumed to be a random sample of the relevant distribution effects, and the combined effect estimates the mean effect in this distribution. Note that in this model, we still have the sampling error, but now we also have another source of error, which is the true variation in effect sizes. Calculating a summary effect is certainly useful in understanding how good a model is at characterizing a human condition, or how well a treatment, drug, or intervention performs in a group of animals. In many cases, what can be even more interesting is the heterogeneity between studies and the differences which account for this heterogeneity. When I say heterogeneity, I mean the extent to which effect sizes vary within a meta-analysis between studies. Especially when pooling animal studies, you will find that the number of studies are many and the study designs can vary massively. According to the Cochrane Handbook, statistical heterogeneity manifests itself in the study effects being more different from each other than one would expect due to random error or chance alone. And it is interesting and valuable to explore these significant differences within research fields to inform future research about how to design subsequent animal experiments and about how findings might translate to higher domains of research like clinical research. So variability between groups of studies can be caused by the samples and their characteristics being used in a study, the interventions administered and their way of administration, as well as outcome measure details and general methodology and the quality of this. Investigating this heterogeneity and the influence played by different study characteristics on the observed effect size can be determined by two different methods. First one being stratified meta-analysis with partitioning of heterogeneity. Here we assume that if certain study characteristics are important, then effect sizes from studies which share these characteristics will be more similar to each other than they will be to studies which do not share these characteristics. The heterogeneity is partitioned into that within groups of similar studies and that between groups of studies. For each group of studies, we calculate a random effect size and a heterogeneity statistic called Q. The heterogeneity statistics for each grouping are added together and subtracted from the total heterogeneity to give the residual heterogeneity between groups. We then use a chi-squared test to assess whether observed differences in results are compatible with chance alone, or whether differences between groups are in fact significant. A low p-value provides evidence of heterogeneity of intervention effects, or in other words, variation in effect estimates beyond chance. A useful statistic for quantifying inconsistency is called I-squared, which describes the percentage of the variability in effect estimates that is due to heterogeneity rather than sampling error. Just one thing to note is that the chi-square test has a low power in meta-analyses involving comparisons of small sample size, or where these are few in number, and you should be careful when interpreting your results in these circumstances. Measuring heterogeneity is meta-regression, and this involves a similar approach to simple regressions whereby an outcome variable is predicted according to the values of one or more explanatory variables. The outcome variable in this case is the effect estimate, and the explanatory variables are the study design characteristics we are interested in, which we think may or may not influence the size of this intervention effect. The difference between simple regression and meta-regression is that in meta-regression, larger studies have more influence on this relationship than smaller studies due to the weighting of studies we perform when calculating our overall effect. Meta-regression takes into account one or more study-level characteristics and determines how much heterogeneity can be explained by taking into account both within and between study variants. Both univariate, where we assess the impact of a single covariable on effect size, and multivariate analyses, where we assess the impact of multiple variables, are possible when performing meta-regression. Where explanatory variables are categorical rather than continuous, dummy variables are required. The choice of which method to use to investigate heterogeneity in your dataset is entirely up to you, but evidence does show that meta-regression can be more sensitive than stratified meta-analysis at detecting the effect of a variable of interest on overall effect size. However, meta-regression needs more observations than stratified meta-analysis and therefore should not be considered if comparisons and studies included in the meta-analysis are few. You may choose to use one method as your main analysis and the other as your sensitivity analysis. This can include the reliability of your overall conclusions. When exploring heterogeneity in your dataset, it can sometimes be difficult to interpret your results. As mentioned, for performing stratified meta-analysis, you would normally perform a chi-squared statistical test for heterogeneity. We can interpret this using the p-value that we obtain, whereby if we get a low p-value, this provides evidence of heterogeneity of intervention effects that are beyond chance. This is also true if you get a large Q statistic relative to its degrees of freedom. I-squared is used to help quantify the amount of heterogeneity that we see. It is a percentage that lies between 0 and 100, and as a rough guide, 0 to 25% is considered to reflect very low heterogeneity, 25 to 50% reflects low heterogeneity, and 50 to 75% reflects moderate heterogeneity, with over 75% thought to reflect high heterogeneity. Having said that, the importance of inconsistency depends on several factors, including magnitude and direction of effects, and the strength of evidence for heterogeneity, which comes from the chi-square test. For meta-regression, we get a different statistic, which measures residual heterogeneity, tau squared, and there are several ways of calculating this. We recommend using the restricted maximum likelihood estimate approach to estimate tau squared because it is less likely to underestimate or produce biased estimates or variance. Change in tau squared following inclusion of covariables represents the change in residual heterogeneity, and the variance in the dependent variable, which is accounted for by these covariates, is used to calculate an adjusted R-squared, a measure of how much heterogeneity is explained by your model. Of course, if you're looking at the influence of a list of different study variables on the same dataset, then you are also testing multiple hypotheses on the same dataset, and your likelihood of one of them being significant due to chance increases with each test that you do. This is called a type 1 error, and when you interrogate the same dataset multiple times, you're increasing your likelihood of this type 1 error. Correcting your level of significance for the number of hypothesis tests you are performing can help to overcome this issue. And by altering the p-value to a more stringent value, we can make committing a type 1 error less likely. However, it is also important to know that the more conservative a test is, the more vulnerable your results are to type 2 errors, which is the opposite and is when you fail to identify a true positive result. There are a number of different tests out there that can be used to correct for multiplicity of testing, including unadjusted least square means comparison, Bonferroni correction test, Sheffield test, and a Tukey's range test. In our work, we tend to use Bonferroni correction, which essentially divides the desired significance p-value threshold by the number of tests that are being performed. But note that Bonferroni correction assumes that individual tests are independent, which may not always be the case. Fortunately, not all studies carried out are published, and those which are may be different to those that are not. And publication bias occurs when the likelihood of study being published is influenced by the findings of the study, and studies are not published due to the direction or strength of the study findings. There are 3 different methods that you could apply for preclinical studies to explore the presence of publication bias. The first one being funnel plotting, where in the absence of publication bias and heterogeneity, these plots are funnel-shaped and symmetrically centered around the summary effect estimate. An alternative is using Egger's regression, which is a significance test for the presence of publication bias in a dataset and answers the question whether the study estimate is related to the size of the study. Finally, trim and fill aims to not only identify but correct for funnel plot asymmetry arising from publication bias. It trims smaller studies causing funnel plot asymmetry and uses this trimmed funnel plot to estimate the true center of the funnel plot and imputes the missing counterparts around this new center. One thing to note is that whereas we nested outcomes before our meta-analysis, for exploring publication bias where different outcomes have been measured in the same cohort of animals, we recommend using each of these outcomes rather than the pooled estimate. Otherwise, in effect, we would be suppressing these studies from the publication bias analysis. Completed your review, it comes to reporting your data and your findings. So here are some pointers. It's important to publish all of your data. Transparency is a key stepping stone of science, and many journals and funders now expect any data generated during a study to be made open and publicly available to other researchers. There are plenty of opportunities to share the data that you have collected in open data sharing repositories like Open Science Framework, Figshare, Zenodo, and Dryad, to mention a few. This enables reproducibility and reuse of the data you have collected, reducing research waste and promoting collaboration. You can also increase the reproducibility of your work by making clear exactly what statistical analyses were performed and reporting all of these analyses results to the extent that it could be repeated by another researcher. Otherwise, you might misinterpret your work and mislead other researchers reading it. And remember, science is only useful to your field if it is robust, transparent, and reproducible. Make use of automated tools to help you complete your work quicker, but also understand what these tools are doing and how your data is being transformed. Otherwise, you might be misinterpreting your results and misleading other researchers. Listening, and here are some useful resources to further help you in your quest of performing a systematic review. We at SURF are here to provide you with further help, so if you have any specific methodological questions, please feel free to email us and we will do our best to try and help.